This is the TokuProfile Web product update webinar. I am Carlos Medina, the Director of Product Management here at TokuBio, and I'm joined by Will. And Will, I'll let you introduce yourself.
Yes, hi everyone. I'm Will Chronister. I'm a senior computational scientist here at Tako.
Awesome. And yeah, we're really excited to walk you through the fully redesigned TokuProfile Web interface. And so in this webinar today, what we'll cover is the intro, and I have a little note to self to ask you to please feel free to use the Q&A and add in any questions that you have as we're walking through the demo and as we're talking about the customer project and showing you the features. And then I'll try to answer those questions in real time if I can. And then any other questions will be answered in the Q&A section at the very end. So let's first start with how to access the TokuProfile Web demo, and then we're going to go into answering a real-world customer question using the web demo's features, and then Will lead the Q&A at the very end. So to access the TokuProfile Web demo, you can go to our website, tako.bio, and in the TokuProfile tab at the top, just click on Interactive Web Demo. This will take you to a login page, and you can log in using the credentials we provided here. And so that's demo@tako.bio. And then the password is tokutest1, which you can see here. And check the spelling. Once you log in, you'll land on your account page. And so here you'll see all of the projects that you have with TakeOBio. For the demo account, we have one project, which is our demo project with Dr. Shernan Holtan at the University of Minnesota, identifying immune features in responders and non-responders in a phase 1/2 clinical trial, evaluating a treatment for acute graft versus host disease. Once you enter the project, your landing page is on this project overview page. And what you can see at a glance is that we have kind of this interactive section here. And this is where you'll see all of your data, all of your plots. And then we have this sidebar on the left broken down into 3 sections. First is About Project, which includes project overview, sample overview, and data download. Then the other 2 are gated analysis, where you'll have the features and capabilities to plot and run statistical tests with the manually gated data. And then below that, unsupervised analysis allows you to explore and generate plots for your unsupervised analysis using the unsupervised clustering data. So let's orient ourselves to the project that we're working on. The customer's goal was to identify immune cell features, including the expression of these 2 markers, AREG and ST2, on immune cell populations, and look for features that separate responders and non-responders in their phase 1/2 clinical trial, evaluating this treatment, UHCG, in acute graft versus host disease. They had 3 key questions. In this demo, we're going to go through the first 2 questions together. How does the immune cell composition compare between responders and non-responders? And they're interested in phenotype or population size, state, cell state, and protein expression of these markers. And then secondly, do immune cells expressing AREG or ST2, do their frequencies or marker expression levels correlate with patient outcomes? And so what you can see at the bottom here are the major immune cell subsets that we've analyzed. So T, B, dendritic cell, monocyte, natural killer, and then other T subsets like gamma delta and NKT. And you can also see all of the state markers or functional state markers that we analyze across all of these populations here. And the last thing we need to look into to kind of orient ourselves to this data set is the sample overview. So we can see that they sent us samples from 20 patients, a total of 2 samples per patient. So that's 40 samples total across 2 time points. And the way that we've grouped the samples below is how we can kind of analyze them. And so we can look at baseline comparing responders and non-responders. We can also look at on treatment comparing responders and non-responders. We can also look over time comparing baseline and on treatment within the non-responder or responder groups. For a full list of the samples and the sample metadata, we can view that here. We can also download the data as well. So we are kind of oriented to the project goals and questions and the samples that we're using. So let's return to that first key question and try to answer it using these tools that we have here below. So how does the immune cell composition compare between responders and non-responders? when thinking about composition, I like to jump down to the stacked bar tab first. Here you can see the immune composition for each individual sample, and it's showing the frequency of all of the immune cell subsets that we analyzed in our data set. To kind of order these, we can group by time point and then by response. And so here we can see that now we're looking at the samples at baseline and on treatment, and then within non-responders and responders. And so we can begin to compare at a glance non-responder immune cell composition versus responder immune cell composition at baseline and on treatment. And so looking even quickly here, I can see some differences in these yellow populations, which you can hover over and see are CD4, T central memory, effector memory, naive. And we see even more of these in the on-treatment time point. So let's filter to on-treatment to only show those. And let's also zoom in on the CD4 populations by deselecting the other immune cell populations from the plot. And now with just the on-treatment time point selected and showing only the CD4 T cell subsets, we can really see that there is quite a noticeable difference between non-responders and responders on treatment. So this is great for kind of giving us an idea of what to maybe look into. In order to really begin running statistical tests and comparing these populations more directly, we can jump down into the Immune Differences tab and start with the Population Frequencies tab. So here you can see you can compare immune differences between response endpoints and look at population frequency, cell state, and marker expression. So here I'm just going to, excuse me, select all subsets, and we can see that what we're plotting now is non-responder versus responder at both time points. And I'm going to filter to just that on-treatment time point. And we can see the stats run up here. And to try and identify, so here we have 4 pages. We're looking across all of the immune subsets analyzed. So let's use false discovery rate to give us an idea of what might be trending or significant. So that'll give us a sense of where to look. So let's set this false discovery rate at 0.1. And now we can see all of these immune cell subsets that are trending and tend to trend higher in non-responders. So what are these? They're mainly CD4, central memory, effector memory subsets. We also see some CD8 subsets as well. Going to page 2, we can see other T cell subsets like gamma delta and double positive. So we can see that there are some CD4 subsets trending higher in non-responders on treatment, which addresses the first part of their first question. Now let's look at expression of these markers like AREG and ST2 by going to cell state. So here in cell state, you can look at the frequency of cell types that are expressing certain markers of interest. So here I'll narrow it down to just those CD4 subsets. And let's narrow it down to just AREG and ST2. And then again, we're going to focus on the on-treatment time point. And so here, what we're looking at is the frequency of AREG expressing CD4 T cells in non-responders compared to responders. And we'll set a false discovery rate of 0.1. And we see that there are no trending results here. And so what if we look at baseline? And so if we switch to baseline, we can see 1 trending result or 2 trending results in the CD4 compartment. And so that's the frequency of AREG expressing CD4 T central memory cells being higher in non-responders. We also see that for the larger and broader CD4 T cell group. So this gets to the second part of the first question, which is looking at the frequency of AREG and ST2 expressing subsets. And this seems to be higher at baseline. And the last part of their question was about the actual expression of AREG and ST2 itself, so protein expression. For that, we can go to marker expression, select response. Let's select our CD4 subsets, as well as AREG and ST2, and filter just to baseline. And here we can see the protein expression of AREG and ST2 on these subsets between responders and non-responders. We set a false discovery rate of 0.1. Unfortunately, we don't see any differences that are trending. And we can also check on treatment as well. And while we do see that they are generally higher, they are not trending towards significance. So we've already answered and addressed the customer's key questions about immune cell composition, AREG, and ST2 expression. So now let's use some of these tools to explore and identify other markers of interest for our study. So I'm going to expand this to all populations and all markers. And we've left the false discovery rate at 0.1 and the filter at on treatment. And so what we can see here is a number of markers of immune cell activation, like CD38, HLA-DR. We can see metabolic markers like GLUT1 that are also upregulated in activation, and markers like PD1, which is the checkpoint marker, and TIM3, which is associated with activation and exhaustion. And so there's some differences in activation markers beyond AREG and ST2 within these subsets. And so let's explore that by using the unsupervised analysis data. So for that, I will go to UMAP. So UMAP is a technique for displaying 40-dimensional data in 2 dimensions. And here we can see each dot represents an individual cell, and this is spread across all of the samples in the study. And so first thing I'm going to do is color by response to look for differences between responders and non-responders. And so what we can see here, we deselect and select non-responder. We can see that there are some clusters present in non-responders that we don't see in these responder groups. And so that's here, here, and over here. And so what do these cells or clusters correspond to? To understand that, we'll switch from response to cluster name. And what it's going to do is annotate or assign names to all of these clusters. And so we can hover and see that those clusters correspond to monocytes in this island. And then in the second island, they correspond to CD4 T central memory and CD4 T effector memory subsets. And we can see that there's multiple effector memory clusters popping up here. And so what we would like to do now is really understand what is the difference between these CD4 T effector memory clusters. Why are they different clusters and not just one together? So to understand that, we can go to cluster heat map and click on the CD4 T cell clusters. And in the cluster heat map shows us the expression of all the markers within each cluster for each group. So what we're seeing now are all of the CD4 T cell clusters here. And you can see that most subsets correspond to one cluster, but this T effector memory subset was broken down into 3 clusters, 9, 21, and 18. And so by looking at marker expression, we can begin to understand what differentiates these effector memory clusters and why the unsupervised clustering split them up. And so going along these 3, we can see that clusters 9 and 18 express activation markers like CD38, CD25, and Ki67 that cluster 21 doesn't seem to express or expresses much lower. These 2 subsets also express checkpoint markers associated with activation like CTLA4 and PD1. And so here we can see that these 2 clusters are can be better defined as kind of more activated effector memory T cell clusters. And so we can begin to understand why these activated effector memory T cells might be more associated with non-response in that UMAP. So there is a lot more to explore. There's a number of other features, including looking at immune changes or kinetics over time, comparing group similarity with our principal component analysis, as well as looking at the composition in different ways using our immune box plot tab. But for the sake of keeping this demo down to about 20-25 minutes, I'll stop there and we can transition over to a Q&A led by Will.
Yeah, thanks, Carlos. Yeah, so we can get into Q&A now. If you haven't already, feel free to submit questions and we'll go through as many as we can. I have one in front of me about the difference between the immune changes over time tab and the immune differences tab. So yes, on the web app, we have these 2 tabs under gated as well as unsupervised analysis. And so the difference between these two is immune changes over time is intended to specifically give you the ability to look at your immune subsets over the course of the time points when your samples were collected. So in this case, we have just 2 time points, which is baseline and on treatment. But you can imagine maybe in your own study, you might have 3, 4, 5, sometimes more time points where you have data collected at each time, and you want to sort of visualize what's happening with a given population. So you can do things like connect the time points by subject, and that sort of illustrates the direction of how these subsets are changing. And again, with more time points, this becomes even more useful as you can sort of see how things ebb and flow throughout the course of receiving a treatment, and then sort of your body adjusting, the patient's body adjusting to it, and then maybe another dose and something else changes. So there's lots of interesting insights that can be uncovered by the immune changes over time tab. Immune differences is intended to be a bit more flexible to whatever your project is looking at. So in the case of this one, we have response, which has 2 categories, responder or non-responder. And so we can sort of pull the samples apart and look at that, look for statistical significance between those 2 groups, and evaluate whether there are differences, whether it's at the population level, the cell state level, or the marker expression level. This tab is meant to give us more flexibility for whatever the experimental design is for the project, whether it's, like in this case, looking at response, or you can imagine another project might involve 3 or more cancer types, and you want to see how the different cancer types react to treatment. We can do that. We can look at more than 2 groups at a time, or we can look at, for instance, like a specific mutation. Like, is your subject, do they have a BRAF mutation, or are they wild type? We can color by that and run statistical testing to see if there are differences there. So that's the immune differences tab is intended to accommodate those types of analyses. And it also does allow you to break down by time point as well, if that's something that you're interested in. So yeah, good question on the difference between those 2. And okay, and I have another question about the UMAP plots. Is there a way to view the marker expression on the UMAP plots? And the answer is yes. And so this is actually one feature we didn't get to in the walkthrough with Carlos, but so this is our UMAP plot again. This is the one way to visualize the results of our unsupervised clustering. And if we color by, I believe it's cluster group, Carlos. That's it. This gives us like a very broad idea of what these clusters are. So we have our monocyte cluster, NK cell, DCs, and then if we want to just do a quick sanity check, we could go to the cell markers drop down, and we could select CD3, which is, I believe, somewhere in the middle. Yes, there it is. And CD19, yeah, that's a good one to just make sure our T cells and B cell clusters are expressing these markers, which are of course is central to their identity as T cells and B cells. And then we can also look at, in this case, the customer was interested in AREG and ST2. So we can look at those as well and see how they are expressed across the different clusters, and maybe that will lead to some interesting insights about where these markers of interest are being expressed. So right now the tool allows you to select 4 markers at a time, and we're open to feedback. If you want the ability to do more, we can look into that certainly. But that's the functionality that we have right now, and we hope it's useful for pulling out more insights from this UMAP plot, and sparking new hypotheses about your data or your study that you're working on. Okay.
Awesome.
Great. I think I don't see any additional questions in the Q&A. So I think I'll pass it back to you, Carlos.
Awesome. Thanks, Will. And thank you for submitting those questions. And so, yeah, with that, we'll close. And thank you, everyone, for joining us for this brief webinar. And if you're interested in trying out the demo yourself, you can go to taco.bio /TokuProfile Web demo, or on our website, as I showed at the beginning. You can also explore the panels and immune profiling offerings that we have on our website at taco.bio/panels. And then lastly, if you have questions about the demo, our panels, or any of our other offerings, feel free to book a call with myself or one of Taco scientists at taco.bio/contact. So thank you for taking the time to join us today, and look forward to having you all explore the web app for yourself. So thanks, everyone.